Hello! Thank you for joining me for this exciting conversation about my project, which involves plant vein architecture, drought, and herbivory.
So before we dive into your experiment, tell me a little bit about your background. What got you into your specific area of research?
Whoosh! That is a long story. I'll give you a brief snippet of my background. I thought I wanted to study plants because I really enjoyed gardening and looking at the different plants when hiking. But then I took a paleoecology class, which I really enjoyed, learning about the past climates. So now I'm in a dilemma. Should I study plants or fossils? I thought a summer internship could help answer this question for me. So I looked up dozens of REUs, which is research experience for undergraduates, and I found one. I read that the project was about leaf vein systems and their functions. It really caught my interest more than any of the other programs. And that's the experiment that we are going to talk about today.
What did you do for your experiment?
So we wanted to test if there was a difference in responses to water stress and herbivory for leaves that have different vein patterns. For instance, there are different patterns of veins in leaves. There are branching veins, which is pinnate, and looping veins, which is called palmate. We chose two species of trees that had leaves that were thin enough for light to shine through because we used a scanner to capture how air traveled throughout the leaf's veins.
Why should we care about your research?
You know, this is a question that I have been struggling to answer. More specifically, for plants on the planet, the Earth is going through climate change right now. Temperatures are increasing, but the precipitation is remaining consistent. This causes the water to evaporate more quickly, and the plants need more water because they are dealing with the increasing heat. As plants suffer in drought, they become more susceptible to herbivory. It's helpful to understand what plants are more vulnerable and to understand why. We are trying to understand the importance of these differences in physiological traits. What plants are more likely to survive? Would it have an effect on our agricultural systems? If the temperature is increasing and we are experiencing more drought and herbivory, it would be important to understand the mechanisms of how a plant is able to cope with these rapid changes. There are two vein network systems, the xylem and the phloem. The xylem transports water throughout the plant, and the phloem transports glucose. So during my study, we focused on the xylem vein network system inside the leaf. The xylem starts down at the roots and travels up to the very tips of the leaves. You may have seen the veins when putting a leaf toward the sun, or if veins are large enough, the next time you look at a leaf's veins, pay attention to the patterns. But we only focused on the leaves for the plants, not any other part. Can you guess how the water is able to travel throughout the veins?
Yes, but I'm not 100% sure how the water would travel through the veins.
What's your guess?
My guess is that the water is sucked up from the sun.
Very good. So the sun is essentially the powerhouse for the water. There's not a heart pumping the fluids through the veins, and the water molecules are attached in like a chain. And this chain connects all the way from the roots in the soil to the leaves swaying in the breeze. And the roots take in the water, and the leaves is where the water escapes. And this water chain is connected all throughout the plant. And when one water molecule is lost, another one is taken in through the roots. So the chain is always connected.
How does the water actually escape?
Great question. Through the stomata. This is where the plant takes up carbon dioxide, which is the main building block for the plant's growth.
And what does this mean for the plant and climate interaction?
Well, as the climate gets warmer, plants have more water stress. There is less moisture in the soil and atmosphere, so the chain of water molecules gets tighter and tighter, creating more tension. This causes the lifelong water chain to potentially develop an air bubble. To compare what a plant has to go through when there's an air bubble, which is called an embolism, imagine drinking a straw, soda with a straw, and a hole gets poked into the straw. It gets more difficult to suck in the soda, which is essentially what the plant is experiencing. Do you know what herbivory is?
I'm not too sure what herbivory is, but I think it has something to do with eating plants.
That is an herbivore, like deer and cows, but I specifically pretended to have an insect bite the leaves. So a xylem feeding insects, caterpillars or aphids.
Did you pretend a bug bit into the leaves to create air bubbles for your experiment?
We did. We created water stress for the samples by cutting the branch off of the tree and seeing how air traveled throughout the leaf. Then we simulated herbivory by punching holes into the leaves that we were experimenting on. We punched the main vein, called the midrib, and we also punched the side of the leaf, which had no main veins connected to it. This is called the lamina. We punched small holes because we imagined an insect biting into the vein system. And we scanned the leaves in about 7-minute increments to detect how air traveled through the leaf veins. So the light from the scanner shines differently through air than it does through water. Sort of like when you put a pencil in a glass of water. It no longer looks like a straight pencil, but it looks like it was cut where the water starts and placed a little off to the side. So the water shines through the air differently.
Were there differences that you were able to detect?
There were differences. We saw differences in the 2 species we used in the different herbivory treatments. The colors of the veins portray specific time frames, the purple indigo being earliest and the white being last. The veins that pop up in the beginning are the first embolisms and continues from there. It's interesting that there are still embolisms occurring throughout the scanning process, indicating that there is a way that water is getting to the other veins even after an air bubble had filled that part of the xylem. Our visual interpretation, it seems as if the leaves with the midrib punched reached hydraulic failure quickest for both species, then the pinnate venation reached hydraulic failure after the palmate venation. Surprisingly, the leaves that had the lamina punched had a delay in embolisms being initiated. Does that make sense?
Yeah, that actually kind of does make sense. Do you have an idea of why these differences occurred?
So we believe that the pinnate species is equipped for drought a little better. We used Combretum erythrophyllum, which is a tree native to Africa, and it goes through dry and wet seasons. So if it has evolved to be able to withstand long periods of drought, we believe that there are mechanisms that allow this species to hold onto its water for longer periods of time compared to the red maple species we used for our palmate venation. Plants that are better equipped for drought tend to be better at fighting herbivory because water is a precious resource for them, so they are willing to allocate more resources to plant defense. This could explain the delay for the embolism initiation for the lamina treatment. Then the midrib being punched, it was like having a main pipe punctured, which would lead to a lot of water loss. It was interesting to see that the embolisms occurred in the other veins, indicating that water was still able to travel through the leaf, even with a large hole in the main vein. And if my research interests you, click on the link below to read more about it. Thank you.
Yay! Good job!